Our topic of discussion today deals with the biochemical aspects of vitamin A. All right. So let's talk about vitamin A. But before that, I think all of us already know this that vitamins they are classified into either fat soluble or water soluble types, right? And under fat solubles, you have A, D, E, and K vitamins, whereas under the water soluble types, you have the B complex and C, right? That's more or less the rough subclassification of vitamins. Today, we talk about vitamin A. Now, before we start vitamin A, I'll like to lay down a general foundation in which, or rather a layout in which we are going to study all the vitamins in our body from the biochemical perspective, right? So think about this. All the vitamins, how we will study them is, first of all, we will like to get a general gist of the vitamin as far as what their functions are in our body. Then, after knowing their importance in our body, we will go on to learn about their biochemical aspects. That is, what the chemistry of this vitamin is, right? How it is, from where is it coming into our body? Where in our diet is it coming from? How is it broken down in our body? How it is digested? How is it absorbed? How is it assimilated? And then we will learn about how it is transported onto the different tissues, and after going on to the different tissues, how it works. That's what we're going to do. At the very end, we will list down some deficiency or toxicity, that is, lack or excess of vitamin cases. This is a general foundation, or rather, this is the general gist of how we are going to study all the vitamins. Starting with vitamin A. Today, our discussion is about vitamin A, right? So, why do you need vitamin A? Just a very general gist. We'll talk about the functions in details a bit later, but just to assert its importance in our body, vitamin A is very, very important for four very important functions. Number one is vision. Number two is, you might say, skin and mucosa health. Number three is growth and general development. And number four is reproductive functions. Although vitamin A has some other functions as well, we will talk about those later. This is the general gist of the four incredibly important functions which vitamin A has in our body. All right. So this is just to give you an overview or importance of how important vitamin A is. Now let's talk about what vitamin A is. What is this, chemically speaking? Now we are going on to the chemistry of vitamin A. So speaking about the chemistry of this compound, what we are left with is very interesting thing. All the compounds which have this vitamin A functionality or structure of vitamin A, all the compounds together, they are called as, sorry about that, they are called retinoids. So listen to me very carefully. Vitamin A is not a single chemical compound. Neither is all the other vitamins. Vitamins are a family of compounds, a family of associated compounds which share a similar foundation structure and share functions. All right. So all the compounds, all the family of compounds which share the vitamin A functionality or vitamin A structure, they are called retinoids. Retinoids, on the other hand, they are subdivided into the major categories chemically speaking, which are classified, or rather which constitute vitamin A. So very roughly or very loosely speaking, vitamin A exists as retinol, retinal or retinal, and retinoic acid. Very important. If organic chemistry knowledge prevails, you will immediately spot that all means this is an alcohol, al or al means this is an aldehyde, and oic acid means this is an acid, obviously. All of them constitute the family of compounds known as retinoids, that is vitamin A, which is a family of compounds. What unites this family is a common structure with some functional group differences. Now do you understand how the classification is? I'm giving you a very rough overview. I would absolutely not say that you remember the structure, because it is beyond the scope of a medical student. I think so. But to give you a very overall gist, if this is the main ring, this is how the tail goes, right? With alternate saturations, or rather unsaturations. At the end, whatever functional group resides decides the group. If there is an OH, it is retinol. If there is CHO, it is retinal. And if this is COOH, this is retinoic acid, right? But the general foundation structure, this is called a beta ionone ring, and this tail is sometimes called a polyisoprenoid tail. Now I would absolutely not say that you remember these things, because very frankly speaking, you may just take a look at these things on the night before the examination, because very frankly speaking, I will not be able to retain these informations as to what the chemical structure, what the name of the ring was, what the name of the the alternating saturation unsaturation tail was, polyisoprenoid. These are very minute things, and I do not think they are worth retaining. However, in examinations, it's a different story. Anyways, so the end functional group, that is deciding what kind of vitamin family you're talking about. Is it retinol, retinal, or retinoic acid? That's actually decided by that. But there is another kind of isomerism. As you can see, this is functional group isomerism, right? This is functional group isomerism, right? No problem with that. But there is another kind of isomerism. That isomerism lies on whether the compound, retinol, retinal, or retinoic acid, has trans or cis configuration. So depending on that, all of these can again be subclassified into different isomers. So a variety of isomers can be produced. For example, there is this very, very important isomer of retinal called 11-cis retinal. Alright, then for retinoic acid we have another, these are examples I'm giving. Alright, that does not mean that 11-cis retinal is the only cis isomer for retinal. There are other isomers, cis isomers of retinal as well. But this is a very important one. Why? We'll see to that later. Again, for retinoic acid, we have 9-cis retinoic acid, abbreviating. Again, we also have 11-cis retinoic acid, etc. Alright, so I'm just saying that this trans, cis, this kind of isomerism also exists within the subtypes of retinoids. Retinoids itself, functional group isomerism, all these three, then cis retinal, 11-cis retinal, all trans retinal. This is a very, very important category. So these are isomers of retinal. Again, they are all possible for retinol as well, retinoic acid as well, but I am only listing down the very important ones, which have something to do with the later functionalities of vitamin A. Understood? Okay. Now, let's talk very quickly about the sources of vitamin A. So, when I talk about sources, I won't list down all the, food items and all of that. I just want to tell you that vitamin A has two sources. One can be from plants, and obviously the other from animals. Interestingly enough, from plants, the vitamin A is, we actually extract vitamin A in the form of, or rather, plants contain the compound beta carotene. Beta carotene is nothing but a dimer of retinol. Okay, it is nothing but a dimer of retinol. Now I'm going to assign a symbol for this, because later when we talk about absorption and digestion, there we have to list down this many times. So I'm going to assume, or rather assign a shortcut to this. Let's call this to be ROH. All right, retinol. This is nothing but the dimer of retinol. Again, animals, they have another interesting thing. They give us vitamin A in the form of what we call to be a retinyl ester, which means that in these cases, the retinol is esterified with a long chain fatty acid. Alright, and that is why it has something to do with fat solubility, and that's why it's fat soluble vitamin, right? So it is esterified with this long chain fatty acid, hence in the form of a retinyl or retinol ester, whereas beta carotene is nothing but the dimer of retinol. Now, having mentioned this very information, we are now in a position to talk about how absorption, digestion and assimilation of vitamin A takes place. Let's move on to that. So, what happens in this case is that, let me draw very quickly a GI tract to explain to you guys what's actually happening. So for example, let's assume this is the stomach that we're talking about, right? It's going somewhat like this. So from outside, you're taking in two sources. One is the plant source of vitamin A, the other is the animal source of vitamin A. Correct? Correct. So plants, you're actually beta carotene, right? So beta carotene, it's going in, right? ROH. What's happening is, down the GI tract, suppose this is the beta carotene, R. For example, I'm abbreviating it as RR. All right. Now, listen to me very carefully. What's going to happen here is that in the intestine, all the major digestion and absorption of, vitamins, fat soluble vitamins and all of these, they happen in the intestine. So what's going to happen is, suppose this is the mucosal cell, right? So this will be in the intestinal cell, this will be like this. This will actually be broken down, since this is the dimer of retinol, it will be broken down into 2 retinol, right? So what we'll get? ROH. From this we'll get ROH. Now, ideally, we should have written a 2 here, 2 multiplied by ROH, because this is RR, dimer. But unfortunately, our body systems are not as efficient as we make them out to be. So the end result, what has been observed, is that from 1 molecule of beta carotene, instead of 2 retinol, we get only 1 retinol. The other retinol is actually lost due to the inefficiency of our digestive system. So net gain is only 1 retinol molecule from plant source. But let's incorporate the animal source as well. So animal, from that, what were we getting? We were getting the ester, the retinyl ester, right? So when it came down, it was actually something like this. This being the long chain fatty acid. What happens is in the GI tract, this ester is actually broken down. It is hydrolyzed, and the retinol is given out, and the long chain fatty acid that is actually separated from it. This enters into the, or this is actually absorbed into the intestinal cell, right? This is all part of absorption. This is, I'm showing you the breaking down, digestion and absorption as well, right? So this is actually incorporated in here, ROH again. This R, I'm writing R, but this is actually retinol. I'm abbreviating that as R instead of writing ROH, but this is actually ROH, right? Inside, this is again re-esterified. Again, this is re-esterified with long chain fatty acid. Again, the ester is formed. And at the end, what is done is a chylomicron is actually produced. This is a chylomicron. So if you remember in digestion absorption, you had read about chylomicrons. Chylomicrons are what? They are molecules, lipid loaded molecules, which are essential for transportation of substances, right? So they are absorbed through the lacteal system, the lymphatic system, drained into the blood, and finally they enter the blood circulation, right? So the net end product is the formation of chylomicrons loaded with obviously the lipid part and the fat soluble vitamin, that is retinol, vitamin A. That is why this is fat soluble, right? Now do you get why this is fat soluble vitamin? This is the reason. So this is re-esterification along with chylomicron formation. Next, from here, obviously this is absorbed through the lacteal system and given out at the blood circulation. So in the blood circulation, I'm drawing the blood circulation over here. Suppose this is the blood capillary or whatever, blood vessel. This is given out, right? So ROH, chylomicrons. This is blood vessel, right? Blood. So let me write here blood. This is the GI tract, right? Now, this is chylomicron, chylo. Now what will happen is very interesting. These fat soluble vitamins, a very important characteristic of them is that they can be actually stored in liver. They can be stored in liver. This is not something that water soluble vitamins are essential or capable of. So the chylomicrons are transported to the liver. So this is the liver, right? In the liver, what happens is chylomicrons, they give up the retinol, and in the liver it is stored as the retinyl ester or retinol palmitate. That is, the retinol is esterified with a palmitic acid, which is a fatty acid as we know. So that is, as an ester, the retinol is stored in liver. So if it is stored in liver, that obviously means that whenever any tissue would require retinol, this will be provided from liver. So did you understand the flow chart here? That's how this occurs. Now let's quickly talk about what happens when this retinol is required by a tissue. For example, a very, very important tissue which requires retinol is the retina of eye. So suppose this is the retina, Suppose this is the retina. So what will happen is, if it is to reach the retina, I'm not writing retina because we'll require this space. Anyways, let's write it here. So what happens is, if retinol is required in the retina, all the tissues that require this, they have receptors. They have receptors for retinol, right? How is this retinol transported to retina? Here a very important concept lies. Again, what is the universal transportation around the body? Blood. So through the blood, again, this is given out to the blood. But there is a very important or interesting thing which happens. This time, instead of going as an ester in chylomicron form, it is binded with a protein which is called retinol binding protein. So retinol binding protein is a protein which is actually synthesized by the liver itself. A very important protein. It is synthesized by the liver for transporting the retinol as and when required to the different target tissues. So when it came, this is a very interesting thing. When it came as a chylomicron ester form, stored as a retinol ester, retinol palmitate, given out to the blood circulation, not as a chylomicron, not as an ester, in the bound form to retinol binding protein. This retinol binding protein bound with the retinol will come and it will bind with the receptors. Okay. Then from there onwards, the rest of the functions will be carried out. That is how this uptake of the retinol binding protein assimilated with retinol occurs for different tissues. This might not be retina just. This can be any different other cell as well. All the target tissues. This can be gonads for reproductive function. This can be a skin cell. This can be a melanocyte. This can be anything. All the target tissues, right? So this is a major thing that occurs, right? Now, let's talk about the biochemical role of vitamin A. Since we are talking about retina, let's talk about what role does this retinol or vitamin A in general have to play in the retina. So, in retina, what happens is, there is a cycle going on. There is a visual cycle going on. That cycle is called Wald's visual cycle. Wald's visual cycle. What is that Wald's visual cycle? I'm just going to give you an overview here. We'll talk about the Wald's visual cycle in the next page. Thing is, in the retina, you have, if you remember, the two types of cells. What were the two types of cells? They were the rods and they were the cones. Rods, they had a very interesting pigment called rhodopsin. Rhodopsin, if we look at it biochemically, this is nothing but actually a conjugated protein. It's a pigment, so it's a protein. It's a conjugated protein, which means that it is a protein conjugated with a non-protein part. What is the non-protein part? Vitamin A. I wouldn't say retinol. It's not retinol. It's a different form of vitamin A. What is that? Rhodopsin actually is actually this ball, opsin, cuddled in the arms of this kink bearing structure, that is 11 cis retinol. So up to now, all the vitamin A forms that we were talking about were retinol, but now this is 11 cis retinol. Remember that example that I had given in cis trans configuration, isomerism of retinoids, 11-cis retinal. 11-cis retinal, this actually bears this opsin, interlocked with opsin to form the rhodopsin pigment, right? So in a very general tone, if I am to say, obviously now you can understand why vitamin A is required for your vision. 11-cis retinal, that means it's a vitamer, right? It's a vitamin, it's one of the retinoids. So if you do not have this parent compound, or if you do not have the parent compound from where this 11-cis retinal is coming, which is vitamin A, you wouldn't have this rhodopsin. So obviously you would have problems with your vision, right? So now let's very quickly talk about the visual cycle, the cyclic events that are occurring in our vision when light is coming, bleaching rhodopsin. Again, in dim light, rhodopsin is reformed and the dim light vision is coming back. Which, by the way, I am assuming that you know that rods are meant for dim vision and cones for bright colored vision, right? So let's look at that right now. So suppose you have rhodopsin in your rod cells. What happens is rhodopsin, if it is formed, or rather if it is pre-formed, we have to assume that light conditions are not there. That is, it's dim light. Because if there is bright light, rhodopsin will get bleached. It will get destroyed. Kind of, not destroyed really, it will get broken down. So let's assume that's the case, that this is light coming down. What will happen to rhodopsin? Very interestingly, rhodopsin will break down into a series of compounds which will later break down into the retinal and opsin part. But before breaking down into its constituents, a series of compounds, a series of intermediates, within a matter of picoseconds, they will be formed. What are those intermediates? They are, first of all, batho-rhodopsin, then lumi-rhodopsin, right? Then meta-rhodopsin 1, then meta-rhodopsin 2. These are the intermediates, more or less. There are other meta-rhodopsin 3 is there as well, and there is some precursors as well, but these are the major intermediates that are formed. So to really remember them, what I use is their acronyms, that is BLM, Black Lives Matter, or beta-rhodopsin, lumi-rhodopsin, meta-rhodopsin 1, 2. All right, 1, 2. This is more or less the important intermediates. Now what actually occurs is the opsin is given out. The ball which was being born in the kink or the arm of 11-cis retinal, that is given out as opsin. Opsin is a protein part. But very interestingly, you would assume that here 11-cis retinal would be given out, but no. 11-cis retinal is photoisomerized into all trans retinal. Interesting, very interesting. So this should have been 11-cis retinal because rhodopsin is made of opsin and 11-cis retinal, but photoisomerization of 11-cis retinal occurs and all trans retinal is what we get. Right? Right. This is happening in the rods or the retinal pigment epithelium. Right? Now a very interesting thing occurs. Either all trans retinal is immediately reconverted, this is bilateral by the way, into 11-cis retinal, which would obviously join with opsin to form rhodopsin, and this reaction, by the way, this occurs under the presence of retinal isomerase, and this occurs only in the dark. So this is how rhodopsin is reformed after getting bleached. So it got bleached, broken down. Bright light is there, cones are doing their work. But when dark light, dim light sets back in, rhodopsin is reformed. Reformed. How? All trans retinal, which was the photoisomerized form of 11-cis retinal, that is again isomerized by the rod enzyme retinal isomerase in the presence of obviously the dark conditions into 11-cis retinal. Opsin obviously is already there, joins to form rhodopsin. But this is not the this is the complete Waltz visual cycle. This is the cycle. But there is another long form as well. If you remember, I had said that being fat soluble, retinol or vitamin A, it can be stored in liver. So what our body does is it moves some excess of this form into liver to be stored and to be given out when necessary. So there is a long form of this Waltz visual cycle as well. Let's write that down. So what happens is this all trans retinal, very interestingly, it is actually converted into an all trans retinol. Retinal to retinol. So in organic chemistry, aldehyde to alcohol, that means you are reducing this. So you need hydrogen, and for that you need reducing equivalence, so you need NADH plus H plus, and NAD plus is formed, right? And obviously the name of the enzyme is retinal dehydrogenase, or another name, alcohol dehydrogenase. Retinol, not null. Did I say retinal? No, I'm sorry. Retinol. Because what did I say? Alcohol dehydrogenase. Either you can say alcohol dehydrogenase or you can say retinol dehydrogenase. Are you getting me? Retinol dehydrogenase. This enzyme, very interestingly, since it's an enzyme, it needs some cofactors. Retinol dehydrogenase needs zinc. So that's why a deficiency of zinc can alter or can inhibit this cycle, can actually lead to impairment of this cycle. That's why you need zinc for vitamin A function. Anyways, so that's retinol dehydrogenase, converting that into all trans retinol, which, by the way, in this form it is being transported, by the way. As I'm writing this, this is actually getting transported to liver. In liver, all trans retinol is actually arriving. And in liver, as I mentioned, is this retinol is stored in the form of an ester, which is retinol ester or retinyl ester, all trans retinyl ester. So in liver, now you have reached liver, all trans retinyl ester. This is in liver. This is stored in liver. And this enzyme which does this is acyl transferase. Alright? Acyl transferase. So all trans retinyl ester, after being formed, it remains like that. This is how it is going. Very good. Very beautiful. One day, suddenly, rods, they are signaling the liver that, hey, we need rhodopsin. We need these, all trans retinal. We need 11-cis retinal. We have actually, what? Depleted our source for some reason. We have depleted them, please send. So then what will liver do? Liver will tell, okay, all trans retinyl ester, for mobilization purpose, it will again be reforming all trans retinol. So again, for mobilization purpose, it is mobilized in this way, not in an ester form. It is reconverted back to all trans retinol. Alright. Now, if you remember, I had mentioned that from liver you're sending retinol to rods. So what do you need? You need something in blood. Via blood this will go bound to, very good, retinol binding protein. So this will be bound to retinol binding protein. Now this will all trans retinol. This will actually move on, and what will happen is all trans retinol, this will actually be isomerized into 11 cis retinol. This all trans retinol, now it will be actually converted, or rather isomerized to 11 cis retinol. All right, so we'll get 11 cis retinol. Now listen to me carefully. For this, what you would require is retinol, because both of these are retinol isomerase. So in the rods you used up retinal isomerase. In the, you might say, liver or in the blood as well, you use retinol isomerase. All right, and what are you getting? You're getting 11 cis retinol. A major confusion which at least used to I used to suffer from this is I used to do all trans retinol to all trans retinal. No, first all trans retinol to 11 cis retinol, because retinol to retinal is not isomerase, it is this retinol dehydrogenase. So now you will convert the retinol to retinal, not from here to direct retinal. Are you getting me? First you will convert all trans to 11 cis. For that you will require the isomerase enzyme. Then you will convert the 11 cis retinol to this 11 cis retinal. 11 cis retinal. Here the enzyme will be retinol dehydrogenase again. So retinol dehydrogenase, and the enzyme used, oh, that's the enzyme. And just like here you had a need for reduction potential, here you will give out reduction potential, because this is oxidation occurring, right? And this was a reversible reaction. That's why we are writing the same reaction reversed using the same enzyme, but this will be reversed. So now we will use up NAD plus, getting NADH plus H plus, essentially completing the cycle. So this lower part is happening in liver. To some extent you are actually transferring, when you are carrying this is happening in the blood, and this upper part is happening in the rods, the rod cells and the retinal pigment epithelium, or the retinal epithelium. This completes the Wald's visual cycle. All right. Now, having completed the visual cycle, we are now in a position to look at the other functions of retinol, or rather the other functions of vitamin A, other than vision, other than its use in vision. What are they? Let's take a look. So the other functions of vitamin A, first of all, the next major function that I will be talking about is the gene regulation. The gene regulation. So try to remember, I had said that vitamin A is very important for growth and development purpose. As far as that is concerned, how does vitamin A bring about growth and developmental changes in our body? By regulating the expression of different genes, and hence leading to formation of different mRNA and the translation of selective proteins, which will bring about the different growth or developmental activities. Our job here is not to look at what those things are, as far as the growth or development is occurring, what are the growth changes or what are the developmental changes that are occurring. Our job here is to find out how vitamin A is bringing about this change, right? So how is it bringing about this change? So remember, I had said that retinol, it can be oxidized to retinal, and retinal can be oxidized to retinoic acid. Retinoic acid, this is the major you might say vitamer which plays a role in gene regulation. How so? Different cells, the target tissues, they actually have the receptors for retinoic acid. So retinoic acid, that will actually bind with the receptors, which will internalize the retinoic acid, and hence retinoic acid will reach the cytoplasm. In the cytoplasm, the retinoic acid, it will have cytoplasmic receptors. So there it will bind with the cytoplasmic receptors. This is the cytoplasmic receptor of this retinoic acid. This complex will move on, and in the nucleus, where the nucleic material is there, the chromatin material is there, they will go on to bind onto the hormone responsive elements of this complex. This is how steroid hormones work, by the way. That's why I'm taking for granted that you know already. So hormone responsive elements, their specific response elements on the DNA, they will go on and bind to that, leading to the transcription and formation of those specific mRNA, and then translation to form the different proteins. Right? Now, an interesting thing to note here is that this receptor that I talk about, this receptor, this can be of two types. The retinoic acid itself, this can be all trans, or the retinoic acid can be 9-cis. So remember, these were the examples that I had given in the first. 9-cis retinoic acid or all trans retinoic acid. If this is all trans retinoic acid, then the receptor is denoted as retinoic acid receptor. However, if it is 9-cis retinoic acid, it is denoted as RXR. Instead of RAR, it is denoted as RXR, that is retinoic X receptors for 9-cis retinoic acid. So why I am mentioning this is because this RXR is very interesting in having some joint activity with vitamin D receptors. So this thing, this RXR, this actually forms dimers with vitamin D receptors. Why this knowledge is important is because of the fact that if there is any deficiency of either of these vitamins, the other's metabolism, the other's functionalities will be affected as well. So that's why deficiency of vitamin A will lead to some vitamin D impairment as well, because the dimer formation with vitamin D receptor by the retinoic X receptors, that is impaired, right? So these proteins, the punchline is that these proteins, the different proteins are formed, and these proteins will move on to different functions which bring about the growth and development. Then some minor functions which I had mentioned, the reproductive functions, obviously. So nothing much to talk about in this one. Many different reproductive genes in the gonads, this vitamin A, the different forms of vitamin A, they can have some role to play in their really activation or inhibition or whatever it is. Main thing being the reproductive system that is actually heavily dependent on the retinoids. So much so that there is a clinical part to this. Very interestingly, retinoic acid, if you remember, I had said that the retinoids, they are very, very important for skin development as well. So in case of acne or in case of any other skin problems, dermatological problems, retinoic acid is administered tropically, right? It is administered tropically. However, it is seen that retinoic acid is very heavily teratogenic. That is, it can cause miscarriages. That's why vitamin A is not issued orally or in pills to pregnant women, because this is highly teratogenic. This will cause miscarriages. That's one aspect of the reproductive functions of vitamin A. All right. Again, we have some other functions as well, like immunological system functions. Immunological functions being that the T lymphocytes, they are differentiated. Their differentiation is heavily influenced by retinoic acid. R acid, this helps in the differentiation of T cells to regulatory T cells, and so immunological function of retinoic acid is also there. Also, like I mentioned, or I did not mention this, there is an antioxidant property of the vitamins, there is vitamin A as well, right? So antioxidant property, this is actually employed in anti-cancerous treatments by vitamin A, right? So that's also the antioxidant property of vitamin A that is employed for anti-cancer treatment, right? How this is actually helping, that's actually not known as of yet. Done. And obviously I also mentioned the effect on skin, right? Now let's very quickly know about what would happen if there is a deficiency of vitamin A or if there is excess of vitamin A. For understanding the deficiencies, all you have to do is remember the functions of vitamin A and reverse that. That function will not occur. So for example, in vision, dim light vision is incredibly important, and hence vitamin A is required for that. Deficiency of that, you will get night blindness. Very, very simple. All right, night blindness. And obviously all of you know that dim light vision, that is actually lessened or to a large extent gone in night blindness. Also the dark adaptation time, that's actually increased. Dark adaptation time is the time which is required by the bleached rhodopsin to reform after re-entering into a dim light environment. So that time does exist because all those chemical reactions in the Wald's visual cycle occurred. So a bit of time is required for those reactions to occur and rhodopsin to reform. That is called the dark adaptation time. In case of deficiency, obviously that dark adaptation time is increased very much. But here is the very important thing. Not only is dim light vision impaired, normal vision is also impaired in case of vitamin A deficiency because of a condition called xerophthalmia. In the name itself, it's given. Xeros means dry, ophthalmos means eye. So xerophthalmia means dry eye. What's occurring is, if there is a vitamin A deficiency, this can actually lead to dryness of the conjunctiva. The conjunctival lining that dries up. Also the corneal covering, that also dries up. So if that dries up, what would happen? The condition is called xerophthalmia, right? Now, xerophthalmia leads to some other conditions. Dryness of conjunctiva, corneal covering, then lacrimal drying, all of these leads to dry eyes, but they themselves lead to some other conditions. What are they? The other conditions are, first of all, xerophthalmia leads to a condition called Bitot spots or Bitots spots. What are they? I just now mentioned that the conjunctiva, it dries up. So what happens is, not only does it dry up, it becomes wrinkled, and at some places it becomes very thick because the water has evaporated. It's very dry. So our eyes, they themselves have this intrinsic wetting mechanism by the lacrimal duct and the lashing of the eyelids. That itself is not working very well due to the deficiency of vitamin A, which has led to dryness of conjunctiva and thickening of conjunctiva, leading to whitish triangular plaque called Bitots spot or Bitots spots. Alright. Another condition which xerophthalmia leads to, that is, I just now mentioned that the corneal covering, the covering of cornea dries up. So that also, if persistent for a long time, if this xerophthalmia is persistent for a long time, then due to the corneal covering drying up, the cornea will soften up. It will soften up. That condition is called keratomalacia. Keratomalacia. So see how all of these are linked together. This is entirely one point and night blindness is the other point. So if this persists for a long time, then cornea, which was drying up, now it starts softening up, and what happens is, when the healing starts, then fibrous or scar tissue that starts developing in the cornea, and this will lead to what we say to be opacity and permanent blindness. So all of these, they will ultimately lead to, if persistent for a long time, will ultimately lead to opacity and permanent blindness. And the steps I have already mentioned: xerophthalmia leading to Bitot spots and keratomalacia, which actually nothing but the cornea softening up, right? And later bacterial infection, that is actually leading to bacterial infection and inflammation, which later, when heals, fibrous or scar tissue that develops in the cornea, leading to opacity and permanent blindness. That's the entire vision part deficiency of vitamin A. But as I have mentioned, there are some other some other functions of vitamin A as well. So what were they? Skin development, or rather skin maintenance. So if vitamin A is deficient, what you get is a very important condition called hyperkeratosis. Hyperkeratosis. Right. Hyperkeratosis, it's nothing but wrinkled or hyperkeratinization in the skin, which leads to the skin becoming very rough, right? And there's a very particular look to that kind of skin, right? So that's hyperkeratosis in the skin. And the most important skin development anomaly, very frequent in teenagers, acne. Very, very common, right? This is due to hypersebaceous growth, due to hypersebum production, right? So acne it not only has a vitamin A triggering mechanism, it also has some other hormones as well. That's why I said teenagers face this, because hormonal changes that lead to acne. But vitamin A also has a role to play in that. And as I've already mentioned, in growth also, vitamin A has a role to play. So deficiency of that will lead to retarded growth. So decreased growth and mental development. That is also very, very interesting, or rather very important. These are more or less the deficiencies due to vitamin A. Now, the last thing is, what if there is toxicity? Toxicity or excess, that is hypervitaminosis. Hypervitaminosis. If that is the case, then not much is actually occurring. Some of the conditions, they are that the skin that becomes a little or a lot dry, it becomes very itchy. There is a generalized problems with the GI tract, a nausea, vomiting tendency. These, all of these occur. This is due to vitamin A toxicity or hypervitaminosis. Right. This is more or less all about vitamin A. So hope you found all of this a little bit helpful. Hope, and thank you for watching. Thank you very much.